Check 1, 2, 3, or howdy. Is it working up there?
Sounds good, boss.
Okay, good. Okay, great.
Howdy, everyone. Welcome to the last seminar for this semester series. And last but not the least, we have our department head, Dr. Dhingra, talking to us today. Dr. Amit Dhingra is the head of Department of Horticultural Sciences and professor of genomics. He completed his BSc in botany from New Delhi, India, and his master's also from India itself. Then he did his PhD in University of Delhi and Rogers University under the fellowship of Rockefeller Foundation. After his postdoctoral training at Rogers University, University of Central Florida, and University of Florida, he joined Washington State University. In Washington State University, Dr. Dhingra's program worked in important biological process that translates to crop improvement. Dr. Dhingra, being a researcher, is also quite an entrepreneur. He has his own startup, Phytolegence, as a spin-off from his lab in 2011. And then he has recently started a new startup called New5, which works on agriculture energy solutions. He is also a chief consulting officer for a startup called Mulek Science, which works on a plant-based alternative protein production. Dr. Dhingra has more than 79 high-impact journal articles, and he has also 6 patents, US and international patents in fruit ripening. So we'll hear more from Dr. Dhingra himself. Welcome.
Please give me a minute. This is my test. All right. Howdy. Howdy. Well, it was 2016 when I learned this tradition in this very room when I came to give a seminar. And one of your students, Taya Brown, taught me that. So in 2016, Dan had said, hey, we'll be looking for a department head. You should apply. Well, sometimes those words become real. So if anything's going wrong, it's Dan's problem. So thank you very much for this opportunity to share my research program overview today. I've been here since last September. The time has flown, and I'm extremely grateful to be here. Of course, first few months, I was very confused. What am I doing in Texas? And now I see the purpose, the mission, and thanks majorly in reason as you all. See, I've learned my you all as well. So very grateful for that. So today, the purpose of this presentation is to give you an overview of the breadth of research that I'm doing, partly to engage in collaborations, as well as to kind of create some interest in my colleagues, as well as students who might be interested. And I'm also available to serve on some committees. That's my way of staying involved in research with you all. So my position is both, it's 50-50 split in administration and research. So I'm in the process of setting up my research program as we speak. So that's one thing I just wanted to say, that the purpose of this seminar today is not to go into depth about everything. It's to give you a broad strokes overview all the way from some of the 2 major projects that have been continuing developed at WSU. And then also some of the additional projects. In addition to that, how research has led to these public-private partnerships, which has opened up many other avenues, not just for me. At Washington State, I've brought in my other colleagues because I can't do everything. I don't know everything, even though I want to. All of us do. But so this is an opportunity. This is sort of a playing field there that we can collaborate and develop from. So with that context, I will jump into my presentation. So of course, you all know that if it works. Oops. Well, sorry. Yeah, that's right. There we go. Thank you. So I have multiple affiliations. Of course, I'm full-time here at Texas A&M. I also stay affiliated as an adjunct at Washington State because my research program will continue till the end of summer in Washington State because of the grants and graduate students. Again, I'm very grateful to both these institutions that allowed this transition. Both sides were very welcoming about this. And I learned that, of course, I'm not the first professor to make these changes. So universities, this was a new learning for me, that when professors leave, if you really want, you can make these arrangements. So, but it takes that leadership, and I'm grateful to both locations. My other affiliations include NuFi, which is the startup from my WSU research program. It's a genomics-based plant production program. And we've recently acquired Ag Energy, which is basically a company that converts biochar to biomass through some biomass processing units. I'll briefly speak about that. Then I've also got involved in Molex Science, which is a company that deals with alternative proteins. And then as a post-harvest collaboration, we started working, supporting a company called Ripe Labs, where we have been funded through USDA, SBIR, and NSF STTR grants, which the companies lead, but we are partners in that. So, of course, disclosure is that there is a conflict of interest, financial interest, and universities allow you to disclose those. And I do maintain that both at WSU and Texas A&M. So my program's mission, the title of this talk, is to sustainably produce nutritious food in abundance. And I've been kind of thinking a lot about this. I didn't start my program that way. But some of the motivations growing up in India, seeing some of the food shortages personally, seeing a lot of folks who did not have access to food, including myself standing in ration lines quite a bit, had a lot of influence in we are all sum total of our experiences. So I feel that that's what has been the motivation. And these problems of changing weather patterns, we've just had a big freeze in the Pacific Northwest, which destroyed a lot of bloom. We don't know what the end result will be. We also know about heat events. You all know that. We need to really feed a growing population. How do we get food? Water is getting limited. Arable land, we're losing 30 football fields every year. I guess it's every day. It's just not feasible. But this is not new. This was also in 1970 when they were saying that we have no resources available. What it means is it's a constant problem, and that's why we need to constantly keep working on addressing these issues. And as we have more technology, people, we need to fuel this planet and continue. So some of the research projects that were developed or have evolved in my research background come from that, these type of larger goals and perspectives. So some of my associations with companies also kind of feed into that. I will try to address the sustainability piece, the nutrition piece, and abundance piece during these titles. So I have a program in plant genomics and biotechnology. Again, the mission is to sustainably produce nutritious food in abundance. That's kind of the goal. And I work mainly in fruits, of course. That's why we are in horticulture, although my PhD was in rice. So any graduate student thinking that they will stick with what they do today, wake up and smell the roses, because we're in horticulture. This is a tough crowd. It's like Thursday evening. You should be, cracking up. All right. All right, what I'm trying to say is those are just training grounds, and then you kind of develop your program. So I won't speak about all the projects. I'll briefly talk about others, a few select ones. Of course, I can't do this work alone. I always like to acknowledge and want to acknowledge my team. I stand here because of them. And I really want to thank all of them they prepared me through various experiences as a professor, as a mentor, as a teacher, as a researcher. So I'm here in front of you today. And a lot of collaborators across the years. Now, my fledgling research program has taken root, another pun. And so we do have some lab members. Of course, Andy, there's a joke or reality, I don't know, Andy. I moved all the way from WSU here to mentor him. If you want to learn more about it, he can elaborate on that. And then we have an undergraduate researcher, Katie Toomey. Is she in the audience? No, okay. So she's working on pecan tissue culture, and we just started the first set of experiments last week. So that's pretty exciting. And joining this fall would be 3 more members moving from Washington State University. And I could not start my research program without the support of Professor Patricia Klein. She's generously provided lab space, because as you know, my lab space probably will get done by this fall, maybe early spring. You didn't tell me that during the interview. So now I'll jump into these conversations now. So I'm going to speak about primarily some of the thematic research about post-harvest quality, pear ripening, and then briefly go into the plastic biology project that has a lot of ramifications as well as some additional projects. So everybody understands over here, or at least is familiar with the climacteric versus non-climacteric ripening. The simplest piece is that in climacteric ripening, typically you have a respiratory burst as ethylene goes up as well. Apples, tomatoes, bananas are classic examples. And then on the other side, non-climacteric would be grapes, for example, sweet cherry, some of these examples, or citrus are some of those examples. But this is not the true story. It's a spectrum. It's not like very siloed processes. There are so much diversity because most of the initial research was done on tomatoes or citrus. We created these two silos, but it's truly a spectrum. And I got to work with Pyrus communis, which was an anomaly in ripening. It's in the same subtribe as apples, so a very classic climacteric fruit. But pears are very unique. They are picked mature, but they're not ripe. The starches are still there, especially the European pears I'm talking about. So this might be an evolutionary piece that's always integrated in some crops where they have to be exposed to cold before a process, the next physiological process begins. For example, dormancy is one of them. Flowering in wheat is another one. So these are some processes that are evolutionarily conserved at different stages, all because the plant wants to be able to disseminate its seeds when the conditions are ripe or conditions are right. So this duration of cold is genetically ingrained depending on what variety you work with. Some of the varieties that we grow in the US were released during the French Revolution. Yes, you heard it right. About 250 years ago, when the French, that's when some of the varieties that we grow, especially this D'Anjou pear, the winter pear. So this is really ancient, and that also tells you that we haven't done many genetic improvements or breeding to weed out some of these issues. But in a way, this is a natural anomaly that exists from where we can kind of learn. Another piece is that in the 1990s, the apple market was going down completely because apples could not be stored very well. You must be wondering why I'm talking about apples when I have a slide of a pear. Just be with me for a second. That's when a molecule was discovered at North Carolina State, 1-methylcyclopropene. It did wonders for apple. 1-methylcyclopropene binds the ethylene receptor, and it prevents further degradation or ripening in apples. Now, apple growers just don't grow apples, they also grow pears. Now, you can actually store apples year-round in controlled atmosphere. So the idea was, hey, let's apply to our pears as well and store them for 1 year. Well, the challenge is, the difference is that apples are picked mature and ripe. Pears are picked mature and unripe. So once you apply 1-MCP, no matter what you do, the pears do not ripen. So we started working on this issue, these two issues about what is the genetic mechanism behind this cold regulated ripening or cold induced ripening. And the other part was, what is the, how can we unblock 1-MCP? This was the holy grail of unblocking this 1-MCP based blocking of ripening. So pears, a great anomaly. We were able to utilize this natural system to study what's going on. So one of my first graduate students, Chris Hendrickson, did RT-qPCR because that sequencing, RNA sequencing was not so common at the time. I'm dating myself a little bit. It's only 15 years ago, but the technology has moved so fast now. So we were able to look at about 130 genes associated in one way or another in ripening. We had just sequenced the pear genome, so we were able to pick the primers to target pear genes because not all tomato genes can be amplified, or not utilizing primers from tomato genes cannot be amplified. So we were able to identify something very unique in that process that we found, surprisingly, that when the pears achieve the cold accumulation that they need, interestingly, the alternative oxidase transcript reaches a maxima. Oops, sorry. Of course, I had to do that. There's no pointer here, right, Dan? Okay, yep, that's. Okay, it's too feeble, don't worry about it. I'll just use my. So what we found out when the fruit had reached 100% conditioning, that means it had accumulated all the cold it needed. The AOX transcript was at its maximum at the time. This is very critical because in all other reports, the expression of AOX reaches a maximum either at ripening or at climacteric, or most likely post-climacteric. The reason is that AOX gives an extra push to degrade the fruit in the end so that the seeds can be disseminated. So this observation opened up some doors for us. When I gave a talk here in 2016, I had talked about this and left some suspense, how we are able to work with this, but today I will reveal that as well. So the alternative oxidase respiratory pathway is known to be induced by cold, but we didn't realize that after it has reached the maximum, it starts going down. It's done its job, it's triggered some processes, and it's probably providing additional electron flow through the electron transport chain in the mitochondria. And it's able to also mitigate the reactive oxygen species that are generated in the core. So this is what we started working on. And some of the publications, if you're interested, are annotated on these slides as well. So our hypothesis was that this expression of AOX is truly critical for cold-induced ripening. And this would also tell us what other genes are getting activated in this process. And we were truly interested in all the genetic elements that are involved. So I'm not going to go into each gene right now. I'll give you a summary of that. What we found out was very interesting. What we found out that depending on the cultivar, different alleles that participate in the vernalization process. Now, when you think vernalization, you think wheat flowering. You don't think fruit ripening. However, if you think of the ontologies of a grain and flowering, these are the same organs. And so what we found out that all these alleles related to endodormancy release were upstream or getting activated during the same time alternative oxidase was going on. However, we feel that this is secondary. And because of these allelic differences between the two, we believe that we have a system which is able to accumulate differential amounts of cold. So for example, a Bartlett, a summer pear, only requires 15 days of exposure at 1 degree Celsius. Compare that to Passek Croissant, which is a 90-day requirement. Angios is 60 days. And then we've got Comis pear. I don't know if many people have tried that. It's a very Oregon niche production area. Comis is only 30 days. So if you really want good pears and you get them green and hard, put them in your fridge for a certain time, because that'll be 5-6 degrees Celsius in your fridge. You can actually just leave them for a couple of weeks. You don't have to leave them for 60 days. It's very interesting. You can reduce the number of days up to 10 degrees Celsius. If you go beyond that, they don't ripen. It's a very unique process and a very unique system. Fascinating. Anyway, so we were able to identify that AOX is really the piece in this that is triggering the initial process. We have also indication that there was probably some bioenergy limitations for the fruit to be able to produce its own ATP and trigger a lot of downstream processes. And somehow AOX, we know that when AOX is activated, it will activate the respiratory chain and produce ATP, which can then provide the energy to do all the metabolic processes to break down starches to sugars and activate NOPGs, et cetera. So that was a pretty interesting, and this part of the project or this observation in parallel triggered another thought. When we apply 1-methylcyclopropene to the fruit, which completely blocks ripening, can we overcome the ripening blockage from 1-MCP? So 1-MCP, I just gave you a story about apples. This really changed the apple industry quite a bit. And this was the Wenatchee lab, USDA Wenatchee lab, which researches Apple, really started applying that. And there's a whole company, AgroFresh, that came out of it. And they've been applying 1-MCP quite a bit to the industry. Now, 1-MCP is obviously utilized in flowers as well, in cut flowers. Anything that you wound would release ethylene, so you can actually block that. So just wanted to say that it's not just for fruits. 1 MCP is already utilized in flower production, keeping them fresh as well. So another interesting thing I wanted to just share with you, I was speaking with a weed scientist, weed meaning the weed, agronomy weed, not the legalized weed or illegal weed. Just want to make sure that distinction is clear to everyone. Let that sink in. I was informed that a lot of the herbicides are conjugated with 1-methylcyclopropene. Can anybody take guess why? Okay, no. So 1 MCP will block any ethylene response. So when you provide an herbicide, it's a stress, which will generate ethylene. A plant can actually, the effectiveness of herbicides can be reduced if it's only the herbicide, because the plant tries to fight it. But if you have already disarmed it, that there's no ethylene being produced or sensed, I mean, it can produce ethylene, but it will not be sensed. So now the herbicide becomes even more effective. That's how you can reduce the amount of herbicide that you need to apply. Isn't that so cool? I was like blown away when my colleague Ian Burke in Crops and Soils at WSU told me that. I'm still fascinated about that. So the idea was, can we block fruit ripening with 1-methylcyclopropene? And is there a way of reactivation? So when we started doing this research, several labs were working on reversing the effect of 1-methylcyclopropene. And the idea was it's all because of ethylene. We had already seen that ethylene pathway, which is quite secondarily or further downstream, is not the issue. It's a primary metabolic issue, maybe an energy blockage or the blockage in the bioenergetics or availability of ATP. So the question was, can we stimulate AOX from outside and reactivate ripening? And this is one of those experiments as an underdog assistant professor who doesn't know what they're doing. So we try to do new things. So we applied 2 compounds that are known to activate alternative oxidases. We applied hydrogen sulfide, which is dangerous, by the way. So we had to go through proper training. All credit to Chris Hendrickson, who got the training to deal with hydrogen sulfide, actually generate the hydrogen sulfide in liquid. And then we also applied glyoxylic acid. And the first experiment we did with hydrogen sulfide, it was fascinating because after the treatment, when we came back to the lab, there was pear aroma. I was like, are the pears ripe? They said, yep, they've ripened. We ate it and we lived to tell the tale. So that's how this research got done. I hadn't shared what those compounds were in 2016, but since then we've been awarded the patents, et cetera. So we are not continuing with hydrogen sulfide because it is dangerous. We actually work with glyoxylic acid now and are continuing this research further. So this is also, I just wanted to show you the evolution of methodology. So Chris did the risky hydrogen sulfide SOCAN project, but farmers told us that you might be activating anaerobic respiration. So we had to come up with a different method. So Shauna comes along, she develops the ultrasonic humidification method. Now this is a little bit of a tricky method, but then Evan, who's a current graduate student, has evolved the method further. He bought some chambers. We got grants during the process, so we had some money to kind of upgrade. I wanted to highlight this to grad students. Your work doesn't stop when you leave. It evolves. Here's an example of one person passing on the baton to the next. So don't think like my work, nobody's going to care. This is a continuation of things. I just wanted to highlight that point as well. So with this treatment, we've been able to ripe the fruit, 1 MCP treated fruit regularly. We can actually reduce the firmness, increase the soluble solids. And the biggest indicator of ripening is internal ethylene, especially in a climacteric fruit. So we definitely see that quite a bit. And also citric acid goes up. We know that when we apply glyoxylic acid, it either activates the glyoxylate cycle or the TCA cycle. And both of them have citrate and isocitrate in that cycle. Of course, when we apply the compound glyoxylic acid, we see a big increase in AOX, which then dies out, but the fruit can ripen very easily. Pretty fascinating, right? Simple metabolic process. So now, of course, we've got a model. What is glyoxylic acid actually doing? Is it going and activating the glyoxylate cycle, the TCA cycle? How is ethylene being produced? So some of these questions we are starting to identify now. I wanted to say another point. The beauty of being in horticultural sciences is that when you go talk to the farmers, there are so numerous questions that remain unanswered. We started with an applied question. We've come down to some metabolic questions that we don't know the answers to because these are not available. These can't be tested in model systems. We have to work in these systems. And now Evan is working on how is GLA working? His question is it a metabolic intermediate? So he's using stable isotope labeled glycolic acid, or is it a redox signaling that is really activating the process? So we are trying to decipher this next process going forward. Well, we've also been able to ripen tomatoes earlier with the glycolic acid, much faster than the controls. When we block ethylene production through cobalt chloride activation or insertion, we are able to block it completely. It doesn't ripen, but definitely we're able to ripen the fruit pretty quickly. We've been able to work with a mutant tomato, the Rin tomato, and we've been able to help develop a pigment, which is very unusual because Rin mutant in tomato does not ripen no matter what. So this is very interesting, and it has a lot of bearing on some of the current project that Andy's working on in ripening processes. We are collaborating, of course, and with the Hagler fellow, Dr. Giovanni from USDA, Boyce Thompson Institute. And so you can kind of say seamless, what we learned in pairs, what we are working with in ripening in this area is continuing. Just want to keep track of time. All right. We've also done some work in, this is a back of the napkin experiment sometimes you do. We were able to ripen grape berries, immature grape berries by application of glycolic acid. And this is where, as we talk about these projects during driving with, for example, Justin one day is like, hey, this is what we've observed. This is where conversations start to happen. So we are exploring some projects. Can we apply this to help our farmers ripen their berries at one time or synchronize? So these are some experiments we'll start doing as well. We've taken this concept and we are producing fresh sliced pears, partnering with the industry. So again, we went from a very applied question. Now that we're able to ripen it, we are able to produce this, this product as well. And it's pretty cool. We get to eat what we work with as well. We don't have to cook it. We've been supported by 2 specialty crop block grants to continue this work. Of course, this work wouldn't be possible without all these folks. I just want to acknowledge Chris, Shauna, Rishi, and Evan, who really contributed to all the publications and patents in here. So now switching to plastid biology, that has been, so I got fascinated by plastids at the age of 14. Everything happened to me at 14 somehow, because that's when you kind of like start observing, or maybe I'm slow. You guys probably started at 7. So what was really unique was I learned about oxygen evolution. I was like, hey, that's other part of our lungs. This must be the most important thing. So I told my parents, I don't want to be a medical doctor. I want to work on producing better food. Of course, they were not happy, but I'm very happy to do this. And also, if I may make an argument, the entire life as we know it exists because of plastics. You may disagree with me. We can have that argument after the seminar, but I stand by it. So the beauty of it is that we don't know about plastids in fruits. We mostly know about it from corn or tomato or pepper briefly. We don't understand the diversity of plastid biology and metabolism. So this was one of those long-term thematic projects I started working. But one of the challenges was, while you can take a leaf and extract chloroplast very easily, you just can't do that with fruits. That was a big defeat for us when I started my program. I thought I have done chloroplast isolations all my life. This will be a piece of cake. Guess what? Nature always beats you. But it also inspires you to find solutions, and I'll share that story. So I don't know what happened there. We actually started characterizing the various stages of this fruit. We wanted to look at how do plastids look like when the fruit is developing. And this question was truly inspired by reviewer number 2, seriously, on an NSF grant, who basically said, why are you working on plastids in fruit? You should work on another system. Well, I didn't work on other systems. I was adamant we wanted to understand, are the plastids really different in apples, or are they same as tomato, right? That was the great question. So when we did the microscopy, we found out that between different genotypes or cultivars, Golden Delicious is carotenoid and xanthophyll predominant. Those pigments are present in the plastids. Granny Smith, which is green, chlorophyll predominant, again, that pigment is in the plastid. Top red delicious, or anthocyanin predominant. What about plastids in there? We don't know. I'll share with you shortly. But what we found out that when we look at the column cells or epidermal cells, the plastids were so very different. And they were not one type or another. They were like very complex. They did not fit the silos that we have come to learn about. So these are the typical tomato ones, and that's not how these look like. And when we started characterizing it across the different systems, we started looking physically, being able to identify different morphotypes within the same cell layer, or the same organ had different morphotypes or different types of plastids. So that was pretty cool. At the time when we were not able to isolate plastids, we took a bioinformatics approach because we had sequenced the apple genome, the pear genome, cherry genome, and we had just started looking at these multiple genomes. We wanted to look at the plastid targeted proteome to complement the observation that plastids have different morphotypes. So we looked at two different methodologies to identify plastid targeted proteomes from the genomes. Now that comes with a caveat. The caveat is that the genomes are in a draft stage. They are not perfect. As you all know that we just completed the human genome after 22 years, just completed that. So we are so far behind, but whatever tools we have, we were able to start working with these two different software. One is sequence-based, which will go and read the amino acid sequence and predict if this protein will end up in the chloroplast or the plastid or not. The other one is annotation-based, which means if a protein is labeled as targeted to be the plastid one in tomato, in apple, it might be as well. But both methods have their flaws, and that's what we utilized in our methodology. So we looked at 6 monocots for the genomes that were available, and then 8 dicots. We also included Amberella, which is an ancient species, because we wanted to be able to compare how have these evolved. These are very detailed paper, and sometimes I go to sleep reading them as well, although very interested in this, so I won't go into details. But in a nutshell, we were able to identify a methodology where when we can combine one of each kind of software, we are able to predict very well plastid targeted proteomes that correlate very well with the experimental work that has been done in other systems. So it was fascinating, but we did find many examples where a gene is plastid targeted in one species, it is not plastid targeted in another. So there are many examples of that, not just between different species. We also found, we also analyzed the pangenomes of Brachypodium and Arabidopsis. We saw some differences there as well. So this is a very unique thing. And now if you think back, plastids allow a plant to really interface with the environment. Stresses are sensed there, the photosynthesis happens there. So there's a constant evolution happening in that area or for the plastid targeted genes. This is a subset of genes that is under constant selection and constant evolutionary pressure. So one of our challenges, before I jump there, was that we were all in silico. And one of the first things, one of my first graduate students did was, and including myself, we tried to isolate plastids from apple. We failed. So I also have come to realize that sometimes projects choose people rather than people trying to choose projects. So I had another brilliant grad student, Ryan, try it, didn't work out. And then June came along and she's been able to crack the code. Now we have been able to identify multiple morphotypes, not just from apple and pear, but mangoes, lemon, avocado. This opens up a whole new world, as Disney would say. It opens up possibilities for us to start characterizing the proteome, plastid targeted proteomes, and thereby understand the metabolic processes. And you might be wondering, how does it relate to anything we do? Well, plastids are the site for fatty acid biosynthesis, volatiles, aromatics, a lot of the flavor compounds that you deal with. The nutritional vitamins are produced there. But besides that, all metabolic processes have to go through plastids. There's one or the other step that is dedicated, majority of it. So it's a very fascinating organelle and how it is participating in these complex metabolic processes as you look across different plant species. Now we can open that, and we actually have a proposal second stage proposal under consideration for large-scale biology at PNNL, which is Pacific Northwest National Lab. So we're very excited. What we've been doing for the last 15 years is reaching this conclusion. So again, acknowledgements to Scott, Ryan, and June, who really carried this work together during their PhDs. Once again, one student's work was picked up by the next. And June is actually transferring here this fall. She's already been accepted, so she'll be joining us here. So she'll be part of your cohort. Now I'm going to jump about some additional projects. And these projects were developed because I work with farmers quite a bit. And they were my inspiration to develop a lot of these projects. So these were, I would call them need-driven projects. But that's where a lot of collaborations happen. And I'm going to kind of highlight some of those, like I just mentioned about the one that I'm trying to discuss with Justin. I'll start picking out people in the audience as we are talking about different projects right now. So these are some projects that feed into the, like for example, the ripening work. We created some populations, but that led to developing populations for dwarf pear rootstocks. That is an interest also. When I presented here in 2016, Trish had asked me, would you be interested in doing pecan dwarf rootstocks? And well, we are here and we are talking about that right now. So it's pretty exciting what we're working on. We have a great population of huckleberry and blueberry hybrids. Huckleberry is a wild species of vaccinium. I haven't found a collaboration there, but I'm sure I will. We also have some apple cultivars for cider, which I'll briefly show what we're doing with that. We've been working on powdery mildew-resistant grape cultivars through gene editing, through genetic engineering. So I have some example. We've also gone into DNA-free gene editing so that we can improve our crops without the GMO issues. And similar gene editing and transformation, we developed methods in strawberry as well as in peas. So the rootstock project has already kicked off. We got a population, part of the population, a couple of days ago at the Fredericksburg Fruit Lab. And thanks to Jim Cummins, who's the director at that lab, they've just recently planted half of that popple box that was actually, it came this Thursday. It arrived on Monday, I believe. So we are starting to do that work. The idea is that there is a good demand for pears in Texas. And you can kind of see that although we bred them, started breeding them over there, there might be applications. Because in Texas, we can grow, depending on the region, we can grow all crops, right? So that was one example. And then we've got the apple cultivars for cider. This photograph is fresh off the press. I just got it 30 minutes ago. So Michael and Tim were working in the greenhouse. So we got some of my selections, budwood, that they would be grafting on and growing it here and trialing out. This has also led to submission of some collaborative grants between all the folks here in the fruit lab. So we can really bring this. So these are about 12 selections that are already tested for great flavor, taste, as well as good cider production. These ciders are unique because they retain the pigments during the fermentation process. So they might be healthful because they have carotenoids. I know that's a stretch, but sorry. Okay, I just wanted to also acknowledge the biotech team in the lab. While these folks do other projects, they are at the forefront of doing these things. Fabi is a grad student, Rish is a postdoc. Bruce has since graduated. Rick is also a postdoc, and Nathan is the person who makes sure all our plants are alive. So these are some examples of gene editing in tobacco, nothing new. We've been able to do DNA-free gene editing in tobacco, already established that, and we are trying to extend it to other systems. We also developed this pea genetic engineering platform, which was developed by Bruce primarily. And we've got this is the wild type flower. You can kind of see we are able to, using CRISPR, we are able to completely eliminate the pigment. There's a cool story behind this, why we are doing this, but it's a visual in the flower that we can see. Of course, we do PCR and molecular characterization, but this has led to some other collaborations because pea transformation is not easy, and pea can be utilized to express different types of proteins, in particular animal proteins, and that's one of my collaborations with the industry as well. These are some confirmed edited events in pea already. In fact, since Bruce has graduated and left, Fabi is finishing up this work and we'll be publishing it shortly. We've also worked with Andy Walker's lab. We've been working on grapes I've had a grape program going for the last 13 years. But recently we started doing grape transformation. We collaborated with Andy Walker. We brought his method to the lab. In fact, I was in his lab in January of 2020. That's when we started doing this work. We've been able to adapt his method, meld it with ours. And Fabi has been recently, like as of yesterday, send me these slides where we've been able to demonstrate GFP expression in grapes. And we've also been able to edit out a gene called OMT3 gene, which is responsible for methoxypyrazine production. This is a great one because when I came here and I talked to Justin and Andrea, they both have history in methoxypyrazine. And you must be wondering, what is that? It's the green, it provides the green bell pepper flavor in red wines. So if you can completely eliminate that, it'll be, you don't have to do a lot of secondary processes to get rid of that. It reduces the value. But the other bigger question is, does OMT3 really produce methoxypyrazine? We've not been able to ask that question. So now with this collaboration between Andrea, Justin, and myself, we will have these tools available. And this could be a great graduate student project in viticulture and enology coming together. So these are some examples where we can come together and do these things. Now I'll switch a little bit to some of the crazy things I did. I was working with farmers. They needed clean plants. They needed genomic testing for getting true to type plants, as well as they needed testing for viruses. So in 2011, I started the first spin out, which failed. So I have a $30 million lesson behind me, what not to do. Happy to talk to anybody. As well as then we restarted in 2020, and that company continues to grow. We've actually acquired another company called Ag Energy. So now we are NuFi with a different logo, but that's how life goes on. NuFi is a full life cycle ag tech company supporting the needs of agriculture. And what we have is this validation piece where we use robotics-based genomics, RNA sequencing to detect viruses, also to develop technologies to do genotyping, mass genotyping. Because in seed companies, if you're going to plant certain variety, each plant has to be the same type. You'd be surprised that in horticultural crops, up to 15%, sometimes 100%, it's the wrong genetics. Imagine planting 2,000 trees per acre of apples, and they all have the wrong rootstock. You have to replant it. And the cost of planting per acre of an apple ranges from $35,000 to $55,000. It's a very intensive process. So this brings peace of mind to the industry. Then we've got the propagation process. Everybody knows tissue culture, but we have customized the tissue culture to the genetic background of the plant we work with. And we've actually integrated vertical farming into that process, which has really changed the pace of things. And then finally, the regeneration part, where any biomass can be converted into biochar, which can be turned back into the ground to regenerate the soils. So we've actually been able to get some USDA SBIR grants to develop this technology to bring it to the market, which is allowing us to do field level testing. There is an epidemic in the Northwest, not pandemic, epidemic. The sweet cherries are infected by two diseases, little cherry virus, and there's called a phytoplasma. With this methodology, we do one RNA-seq, we can detect all pathogens. We don't have to do one test at a time because that's the standard currently. So this really provides a great platform. Then the propagation piece, usually when you're trying to produce rootstock, it takes about 4 to 5 years to produce rootstocks. And every season, you can grow a plant by 6 feet, or not 6 feet, but 6 or 10 nodes. We can do all that in 6 weeks by combining the environmental conditions. So this is the multiply process. And we've been funded through this WSDA, or Washington State Department of Ag. So in a way, we're trying to bring science out of the lab directly in the hands of the farmers. One of the motto that we as professors and scientists and upcoming PhDs can do is we have the power of science that we can bear on all these issues that farmers face. So the other part is biochar. In 2015, when I got asked to participate in biochar, I called it snake oil. And the investor who was asking me to work on it said, aren't you a scientist? Aren't you supposed to test it? And that led to a journey where I've kind of gone from a complete non-believer to now I'm going out there and talking about it, because I realized that biochar is not created equal. Crop residue biochar is rich in nitrogen, low in carbon. It has great carbon exchange capacity. It also is hydrophilic compared to wood-based biochars that can be hydrophobic. They also contain tars and oils. So the reason why I thought it was snake oil is because wood-based biochar is available freely. And sometimes it works, sometimes it doesn't work. So we've been able to see consistent results. Just as a brief overview, these first two trials in tomatoes and onions were done by a third party, ag development research firm. They saw an increase in yield in these tomatoes when biochar was applied compared to no biochar. This is crop residue-based biochar. We did an experiment in our organic farm, and we found out that the fresh weight increase ranged from 39% to 86%, depending on the genetic background. So that was another reason why it seems like a snake oil, is because people who were doing research forgot that the genetic background obviously influences the interaction with biochar. So we have another paper, which is on a preprint right now, where we've demonstrated that 3 different types of tomato genotypes and 6 different types of biochar have an interaction, and we see detrimental effects in certain combinations, especially the wood-based biochars. But in the crop residue-based biochar, we see a benefit. These are tomatoes side by side grown in that trial. You will see that on the right. The tomatoes, or we've seen this time and again, I don't know what the rationale behind this is, and we're starting to test that. The plants achieve maximum growth or reach their vegetative maxima faster in the presence of biochar. And of course, we have measured photosynthetic rates. They are the same. So the question is, what's happening in the plant that's making it do this? Sorry, my allergies. Don't worry. While you enjoy the site. So we obviously went the obvious place, the root systems. We really wanted to see what's happening. And of course, the hypothesis is that it's probably microbes. Now, a lot of the work done with microbiomes is DNA-based. I come from an RNA world myself. I thought, well, let's figure out a method to isolate RNA from these roots, which are exposed to these different soils, so we can actually capture the microbes that are associated with the roots. It took us 2 years, because we are slow, to develop a method of high quality RNA. RNA is highly labile, it degrades very quickly. So we were able to find that, and I also like time courses. So we actually sampled the soil or the root systems at 4 different time points. What I want to show you below is just some preliminary data. We are working on finishing this paper right now. Is everything in gray is the control. All these are gray control. The colored ones are 2 rates of application. 1 ton per acre application or 2 tons per acre. But it's not the entire acre that was spread, just the rows where we grew the tomato plants. What is very interesting is what you will note is that at time point 3, which is fruiting, when the fruiting just begins, the microbes reach a maxima and then they decline. I was presenting this work to Rodale Institute, the chief science officer, and she asked me, but don't we want microbes to just keep growing? Well, competition. And that's why in organic production systems, we see decrease in yield. If microbes keep on growing, they are probably competing for nutrients at that point. So in a way, perhaps this is one of the rationale reasons why we see an increase in yields. We're also analyzing the tomato transcriptome, what kind of genes are getting activated as we see the abundance of some of these plant growth promoting microbes going up. So it's a very unique view into this. And of course, I've been speaking with Betsy, who has a lot of expertise in root biology and sustainability, to see how we can build upon this study. With biochar, we are also talking about doing trials with Monty in pecan orchards. In fact, we're working on a big grant right now to submit to the USDA climate change RFP. So these kind of projects, as you can see, are breeding new projects, new ideas where we can collaborate. Also got involved with a company called Ripe Labs. We are funded through, we are able to support their growth through SBIR, STTR, and USDA SBIR grants. It's pretty cool because the funding rates or success rate in these grants is 33 to 50%. Compare that to 3% with regular grants. So we're also able to contribute, companies create jobs as well. So Ripe Labs, what they're doing is they're able to utilize the natural plant volatiles and use them to coat fruit material or post-harvest fruit material to work as fungi static molecules. So we are working on various aspects of this project right now. Of course, it's proprietary, so I just wanted to give you a little bit of an overview. And finally, none of this work would be possible without the funding support that I've received from so many groups. And very grateful to all my collaborators. Again, I'm very grateful to be here among you all. If I missed anybody acknowledging anybody's collaboration, pardon me. I'm just really excited to get started. So I'm interested in collaborations. I'm interested in new lab members. And that was the purpose of sharing this slideshow today with you, or provide you an overview. I'll take any questions. Thank you so much.
Amit, I'll start the question since everybody's thinking.
Could you speak up, please, or can I?
Can you hear me?
Yes.
Okay. So, Amit, it's a great presentation. I really enjoyed it. I have a question. There is a trend now to grow apples in completely tropical countries. So what are your thoughts on that? Because the apple also needs, like pear, some of the chilling requirements.
Yeah, so they've been able to breed low chill apples, or basically have been able to breed out the requirement for chilling. And that's where, as I was saying earlier, in pears, we haven't really bred them for the last 250 years. So we have not been able to do that. But low chill apples have been known for a long time. It's a genetic trait. And so that's how they're able to grow it in tropical areas as well.
Okay.
So we might be able to grow apples here. Who knows?
Yeah. There's a big trend in particularly in tropical countries. And I was wondering about, are there any specific varieties they developed? I was wondering.
Well, yeah, there's genetic material already available to do that work. And the good part is, throughout history, people have eaten apples and thrown the seeds around. All we need to go is find some of those wild ones. There was a cool project we got involved with at WSU. It was called the Lost Apples of the Palouse. People had a lot of apples in their backyards, 100-year-old apples. So I would get calls, can you come and see and identify this apple? And when you visited them, it was not about the apple tree. They would share their family stories. So I went deep into like Idaho, or sometimes in Montana, as well as far as in Oregon. It was a fascinating journey to go and see these big mammoth trees that grow in varied environments. Yeah. Hi, Dan. Yes.
You know, at least one big fortune that's been lost trying to grow apples in Texas. You know what type of rootstock Tim Hartman crashing those apples on?
Tim Hartman, can you respond? What rootstock you're using to graft the apples?
I'm just grafting.
How does that do?
It's delicious.
So,
On that subject, if you were to draw a circle about all the impacts on plant growth, production, and fruit quality, you say soil is a factor, people are a factor, economics are a factor, climate is a factor. All those factors impact how the plant is going to perform.
yes.
But very few people that move to Texas fail to realize we're hot. We can't do dwarf. Dwarf are made dwarf by limited roots. With limited roots, you have limited water uptake. Plants can't make it in Texas with limited water uptake.
That's a great point. And I would say that, again, in my ignorant enthusiasm approach, maybe we can add some things to the soil to retain the moisture, such as biochar, because biochar has been shown to reduce the need for water by 25%. And that's some of the discussions we've been having with Monty as well, to see if we can augment the soils with biochar. And actually with biochar, you can reduce the bulk density of soils, so whereby the clay soils can become more porous and hold on to the water in a bioavailable form. So point well taken. We will empirically test that. As I said, in my ignorance is bliss, so we're going to try something new in that direction. But a great point, thank you. Are the folks online able to hear what is being said in the seminar room, or I can repeat?
We could hear.
Okay, thank you. There's a question from Ben. He's asking about the research around DNA-free gene editing. Could we introduce a little more about that part of the work? So DNA-free gene editing involves taking the guide RNA, which will go and find its gene and edit it, binding it to the ribonucleoprotein. So the CRISPR, the Cas9 protein comes along, the guide RNA comes along, and then we are able to utilize gene gun to introduce that. There are other methods of introducing that, but we've been utilizing the gene gun to introduce the ribonucleoprotein. And because the other challenge is this is very easy, because once you have the mutation, you can see the albino plant, because we are mutating the phytoene desaturase gene that is very important in the photosynthetic functions. So once that gene is, or chlorophyll biosynthesis, when phytoene is gone, desaturase is gone, your plant is albino. The challenge that we have another project in parallel going on grapes, where we are trying to screen thousands of plants through sequencing to identify the edit, because it does not have a phenotype that we can readily identify. So it's a high risk, big number. So I hope that answers your question, Ben. Any other questions? Yes, sir. What was the trick behind getting plastids? So what did we do to isolate plastids from the fruit? There are many variables that we changed. I will start with how the peel was frozen and extracted to centrifugation steps. Usually the chloroplast isolation literature only talks about 3-step gradient. June had to develop 6 to 8-step gradients. And then she was starting to see at multiple levels. And then centrifugation had to be balanced. We also used the freezer mill, which is a way to pulverize tissue under liquid nitrogen in the process. So a lot of these things had to be fine-tuned at each step, which tells you that most unsurmountable problems can be overcome, especially you just need diligence and step-by-step improvement. So all credit to June for doing this. Hi, Dan. We thought that they will happen. They did not. In fact, we are doing some viability tests. We have a collaboration with a biophysicist at the WSU who can check for functionality as well as pigment content. But we've already done FACS analysis, fluorescent assisted cell sorting analysis, and that also is able to capture plastids that are intact, and 90% of the plastids are intact. So now that's where, because of that data, we were competitive enough to be selected for stage 2 proposal at PNNL.
Amit, one more question. Is the hydrogen sulfide, is it normally treated, or I know you probably did it for a lab purpose, but in practical ways in industry, can we use that? And what are the risks involved?
Hydrogen sulfide cannot be used commercially. That's why we stopped using it. It was great to prove a concept that we can activate AOX, and that's why we looked at an alternative method. There are many activators known for AOX, but somehow only glyoxylic acid at its natural pH works, which is around 2.9 to 3.3. The pKa, which relates to pH, is about 2.9 for glyoxylic acid. We've tried succinic acid and other intermediates in the TC and glyoxylic cycle. Nothing else works. So it's a very interesting observation after we learned that we can activate AOX with glyoxylic acid.
Thank you.
The other compound is hydrogen sulfide. Yes. So hydrogen sulfide will block the electron transport chain, which will then force electrons through AOX, and thereby activating AOX. Whereas glyoxylic acid is also, once it starts producing the malate, malate will go back and forth between glyoxylate cycle the citrate, isocitrate will also be utilized in the TCA cycle, as I was showing in my slides. And that's where we feel like it also then activates the mitochondrial respiratory transport chain, which then generates ATP. So it seems like there's an ATP limitation to the process than anything else. And that's why we feel we can activate and other crops and other fruits as well. So I think his hand up went up first.
What's the status you were talking earlier about how they science with the relationship to one what's the status of it? Is it going towards commercialization after that, or I thought that was really
Actually, all herbicides, most of those herbicides that are listed already are being sold. They're just conjugated with one MCP. That was so it's been commercialized for a very long time. Yeah, but a pretty cool concept, right? Yes, Sash. So the question is, can we explain the process of fruit ripening from the outside to inside? I think that's another holy grail. It's somewhat, we don't know how the signaling works, especially in these dense fruits. We don't know that, how the ripening that is sensed or the molecule starts activating outside. There are many hypotheses, right? One of them is that once the organ gets saturated with as a sink, the state-specific genes get triggered, and that's when the ripening kicks on. In this particular model, when we do the labeled work that we are planning to do with labeled glycolic acid, we will be taking slices through the longitudinal section to see how it goes from the outside surface to the internal surface. And that might give us some ideas. Yes, sir.
On that, good point to worry. Which comes first, ethylene and the ripening, or the ripening gives us that?
So respiration comes first. And once the respiratory burst happens, the ethylene is already being produced. There's a system 1 level ethylene that is at a certain level already being produced. There's just a second burst that happens, and that could be because respiration is then really activating the whole process.
So which one comes first?
They all come together. They are synchronized. They're synchronized. They come together.
You're talking about a second product in addition to ethylene that affects ripening.
Yes, I'm talking about.
Which one, does that come from inside or does that come from outside?
That also comes from the inside, the AOX, the alternative oxidase-based secondary respiration or alternate respiration.
So it is there first.
Yeah.
And then ripening.
Yes. If I'm understanding you correctly, my answer is yes.
I don't know about your answer, but I understand. So let's go over it again. Is the ethylene first? Or the ripening?
Ethylene first.
Where does it come from?
The ethylene is being produced regularly through methionine. Methionine is the basic methionine. That's the amino acid. That is the source of ethylene through the Yang cycle in fruits. So Dr. Yang at UC Davis, that's called the Yang cycle, demonstrated that ethylene is constantly being produced across the plant through methionine as being the primary source, metabolic source for that. It's first. Ethylene is always present. Yes, we worked. I didn't show another project that we had where we were able to induce abscission in sweet cherry by ethifon application. What is unique about that is sweet cherry is a non-climacteric fruit, but it has a very specific response to ethifon by creating an abscission zone specifically in one genotype of sweet cherry. So yeah, I worked with ethifon as well. That obviously you can add ethifon to tomatoes that go to ketchup so that you can ripen them very well. Yes.
Is there an agronomic practice to be able to provide assurance for like a tree instead of like an existing orchard that's with tomatoes, you can sell them all, you know?
So the question is that can we put it on perennial crops? That in literature, so we did an article, a review of meta-analysis article highlighting that piece, that there is not enough information available for perennials. However, last season, we applied biochar to some new apple plants that were planted after they had been planted. And we did see in the first year about 20% canopy growth over controls. This was done through an aerial measurement of the canopies. So now the question is, what happens if we apply or don't apply it next year? So repeated application year after year is another uncharted territory for biochar research.
Does it have to be like filled in?
Yeah, it had to be tilled in. Actually, these farmers found a unique way of injecting it in. That is a piece that is being figured out. So now that biochar is part of my company, we are thinking about developing granules out of that or creating different methods of drilling where you can create particles so to improve the application. Yeah. One more question. No, so citric acid going up just means that it's kind of creating the acid balance, because when a fruit ripens, all starches go away, sugars come up, and malic acid, citric acid increase. So that just shows that citric acid is becoming available. It doesn't really change the profile. You can't tell a ripe fruit from an unripe fruit. I mean, sorry, you can't tell a GLA ripened fruit and a normally ripened fruit. Yes, thank you, Andy. And there's another question out there. GLA work, do we know if it targets MCP-induced proteome transcriptome or also acts independently? Well, with the transcriptome work we have done post-GLA treatment, it doesn't really go into the ethylene pathway that much because all the ethylene genes are working fine. What it's doing is it's basically activating some of these AOX-related as well as the enzymes in the TCA and the glycolate cycle. And one more question is, besides the exogenous application of the NOCAN, okay. Did you try another way, for example, to control the atmosphere, keeping the pairs near the low oxygen limit? So yes, these 1-MCP treated pairs, there are about 24 labs across the world who have tried all different parameters and variables. So that's why I didn't repeat those, because everybody had spent about a decade or 2 decades trying CO2, nitrogen, hydrogen peroxide, what else? I mean, you know, when they apply 1-MCP, they would add CO2 gas with it. So they've tried multiple things. So I sat in a conference in Argentina for a whole session one day where I got a glimpse of everybody trying everything. So then the question came, is this even an ethylene-based process? So because 1-MCP blocks ethylene receptor, naturally we tend to think, oh, it must be ethylene, right? We thought, like, forget that part because everybody's trying it. Let's look at something else. Ignorance is bliss, right? Well, this is a wonderful discussion. I think we are already past 5 p.m. Thank you so much. Thank you.